Hello, welcome to the meeting for this week. It doesn't look like anyone's here today. I got a message from Ujwal and Asmit and, oh, there we go, Susan. Hello.
Hi.
Hi. So,
I have to leave early today.
okay. Yeah, that's fine. Welcome.
Okay, I've got a radio in the background. I will hear that I know.
Yeah.
Okay.
Okay.
So I should turn off my there. I don't know.
Oh.
I have trouble with my camera. It's not a very good camera.
Yeah. So I got a message.
Oh well, the fun thing, I got a new deck. If you'd like to see it.
Yeah.
Yeah.
Okay, this is the new ball microscope.
No,
It's very washed out, but it's kind of, yeah, you can kind of see the edges of it. Yeah.
Of course it is.
Yeah.
Anyway, it has 4 screws instead of 3. And there's microscope holders with some screws on them. So that this should be easier to operate.
Okay. Yeah.
Anyways, I went to a fun project.
Oh, that's great. Yeah, great to see that. You're going to hook it up and try it out soon?
Yeah, I need to clean up my upstairs so I have a nice clear table.
Okay.
And then I can do this.
Oh, good. That's great.
Yeah.
So I got a message from Ujwal and Asmit and Tarun saying that they wouldn't be able to make it at this time often, so we're not getting an update from them today on the Basel area. But thanks to them for joining in on Hacktoberfest and working on the Basel area stuff. So it looks like they're making some progress on that. I'm not going to say anything more. They might come and update us in this meeting, but we'll meet separately because it's more convenient for them. But yeah, they're making some progress on the images and segmentation. So last week we talked about how their their choices of segmentation and how they're annotating the images. So I think that's good. I don't know who else is going to show up today, so why don't I go over a couple items and then maybe dive right in, maybe some papers, and then people can watch this online and see, maybe we can, if you want to check some of the papers out, I'll put them in the Slack. So let's go to the screen share here. Sharing my screen. The first is to go over the submissions. And so again, our submissions are, we have a number of things that are outstanding. We have the Boring Billion, the Kindle book. Actually, in the Boring Billion area, I was thinking of a project that might combine the Boring Billion with some of the early embryo stuff we did in a paper back in 2018. It was looking at what we call first mover advantage for embryos. And it's a little game that you, a game theoretic model that you can apply to it. Oh, there's Dick. Hello, Dick.
Hello.
Let me go through this document and then we can, I know you wanted to present something.
Yeah, I can wait to see if anybody else wakes up besides me.
Yeah. So, yeah, this Boring Billion idea is something that I have kind of sketched it out, but I have to, put a couple slides together to explain what it is. This mathematics of DevoWorm, this is still outstanding. This is like a poster of different equations and models that are used in DevoWorm. I was thinking maybe like a short, it could be a presentation, it could be a short paper. I'm not really sure yet, but we still have that outstanding. The test of Williamson symbiosis, which is this, it's a bioinformatics project where you look at different genomes and see if, those genomes are a mixture of different organisms in terms of their developmental program.
We're still hunting for somebody who's interested and has the genomics background.
Yeah, it's kind of tough to find. Well, I mean, not necessarily, but someone who has that specific background. Hello, Jesse. How are you? We also have this molecular level simulations. This is for diatom motion. So we're getting these images processed using the pix-to-pix method. So this is going to be, this will allow us to look at the motion of these colonies in terms of the single cell. So we'll have each cell, and we'll be able to measure each cell independently in terms of their velocity and their acceleration, and maybe some of their higher level derivatives as well. So that'll be maybe something we can follow up on once that's up and running, and converted into new numbers instead of images. The same goes for this Diatom Movement number 26. The quantitative comparison of archaea and shaped droplets, that's kind of dropped off the radar, but this is the thing where... And I think,
no, not really. Deb and I, Mike and I are working on it.
Okay, that's good. That's ongoing. Then there's this book by Stephen McGrew, Eye of Nature. I was reading through it. It's a very interesting book. I don't know what we want to do with it. Of course, that's his post-mortem book, or it would be a post-mortem thing that we would do to honor Steve. So I don't know what that would look like. But that's outstanding. Then we have these different conferences and workshops. We have NeurIPS, which is in December, and then Neuro Match 4, which is in a little bit earlier in December. So it doesn't look like the overlap. I know we were talking about maybe the overlap, but in terms of NeurIPS, if people are interested in machine learning, deep learning, and they want to go to a good conference, this is the Hallmark conference. We're not really involved in that, but we are going to be involved maybe in Neuro Match 4, and I'm still trying to think about maybe a side event that we can do that would, maybe run during this period or near this period, and by drawing people from Neuro Match. And I think Jesse and I have been talking about that a bit, but we haven't really planned anything out. We're gonna kind of have to move on that if we want to do that, because it's already November 1st. So I don't know what your thoughts are on that, Jesse.
I kind of missed what was the subject of your comment?
Oh, about the Neuro Match 4 event, the satellite event that we might have. It wouldn't be like sponsored by Neuro Match, but it would be like kind of adjacent.
Yeah, we should talk about that if it's going to happen. I haven't done anything with that idea yet.
Yeah, I mean, I don't. Yeah, it would require like some content and then advertisement. Those are two things. And then if we get those in place, then we can do we can just say, well, like we did with the other session that we did this summer. We just planned out the content, and that would probably be on me, and then plan out a time, and then advertise it, or get people who are interested in it. And then if they can be like the core attendees, and then you can get other people coming in that once you already have something established where there are a couple people, then people might follow in. So those are my thoughts on that. I don't know, but well, more to come on that, I guess. So Dick, did you have something to present?
Yeah, let's start. I'm going to start with a movie.
Okay. Okay.
Let me share the screen. Share. Come on. Share. Okay.
double sticks.
Can you see this?
Yeah, I can see it.
First, the rigid silicon.
I see. Oh, wow.
Okay.
Centric diatoms are considered immobile. However, some undergo slight shuffling motion. This appears to result from mucilage secretion
Let me start it from the
through a
beginning.
special pore called the labiatum double sticks. Enclosed in their rigid silica wall,
Can you see
diatoms
this?
cannot move like other small cells. However, some species move with a characteristic jerky, gliding motion, here accelerated about 25 times. Here, gliding is shown in real time in a mixture of marine species. This characteristic type of movement is generated by a mechanism unique to diatoms. Centric diatoms are considered immobile. However, some undergo slight shuffling motion. This appears to result from mucilage secretion through a special pore called the labiate process, which may have been the evolutionary precursor of the morphological feature called the wraith. With one exception shown later, pinnate diatoms that glide all possess the wraith, the name given to a very narrow slit through their valves. This pinnularia shows the common morphology of a central wraith running the length of the cell and interrupted with a central occlusion. Two broken valves show how the wraith does go right through the wall, even though the slit is deeply curved. The morphology of the wraith is variable, but it usually follows the contours of the cell. In other species like this Surarella, the wraith runs around one or both edges of the cell. Here is the wraith of a live pinnularia. Viewing it at high magnification and focusing down into the cell, we see active cytoplasmic transport just under the wraith slit. Staining cells for actin reveals two long bundles of actin filaments very close to the cell membrane along the wraith. When actively gliding pinnularia are firmly pinned down, sticky elastic mucilage is secreted through the wraith. It accumulates while moving back and forth outside the cell. So, while mucilage oozes through the wraith, it remains attached internally to the cell membrane. Then this attachment site is moved back and forth by the adjacent actin filaments. In this slender nichia, the wraith lies along one edge of each valve in a narrow tube-like structure called a canal wraith. At higher magnification, the canal wraith is indicated by the arrow. Cytoplasmic particles move rapidly along the canal wraith in both directions. This image is in real time with computer-enhanced contrast. The speed of transport inside the cytoplasm matches that of the mucilage just outside the canal wraith. One sequence is slowed to show this correspondence more clearly. Tiny polystyrene beads sticking to the mucilage outside the canal wraith act as external markers of this transport. In summary, gliding movement is generated by mucilage secretion coupled with cytoplasmic transport. This motile system can generate significant force. When this nichia gets stuck, the cell shows remarkable persistence in trying to move. The force it generates loads up the wall like a spring. This cell persisted in its behavior over 20 minutes. On some occasions, the force generated by the cell can permanently damage the wall. As noted earlier, there is one known exception to this motility mechanism, and it's displayed by the pennate diatom Ardissonia. It has no raphe, but it also moves with a jerky gliding motion of raphed pennates. Most of them double sticks.
Okay. Okay, that's it.
Okay.
Let me show you something else.
All right.
Okay, how many of you have played with bubbles?
I have.
You have, okay. Okay, I want to show you, this is a review, it's now out, that I did on the history of models for diatom motility. I want to show you one picture in here. Let's way back to the end. Let's see, is this it? Yeah. Okay, here's a bubble, and this is after it's drained for a while, so you're getting interference colors. And you can see that the bubbles tend to drain in long, narrow strips. Okay, they sort of channel like a river. Okay, so I use that as a starting point for a model which I developed with Shruti and Krishna. Okay, which we just submitted. And let me see if I can find that. Shruti. Yeah, there it is. Okay. Okay, here's the paper we just submitted a couple of days ago. And I want to show you a number of things about the program. We wrote a computer program. We started in Mathematica, and they got disgusted with it, and we switched to Python. Okay. Now, in doing this program, we needed a number of different variables, all of which are unknown value. Okay, so that means we can do a simulation, but there's no way we can do a simulation that matches a particular diatom and presume that the variables mean anything. Okay, so we just did an example. Okay, so we had, I don't know, about nine different variables here. Okay, so we made a table of our variables and our default values. Oops. Okay, now this was the key to writing the program. The program contains a table. Let me enlarge it a bit here. Oh, I want this enlarged. Let's see.
It's okay. Yeah, I can see the table.
Oh, you can see the table. Okay, fine. Okay, what we did is we assumed that along the raphe there are discrete sites. Now, this is a simple example where there are only 17 sites, labeled 0 and 16. Okay, and each one contains one molecule. Well, I should show you the startling evidence for these molecules. This is a picture of a raphe with these molecules sticking out, and you can see they look like they're in a one-dimensional row. What can I say? This is this is the only picture of this type I've ever seen. But it suggests they're in a one-dimensional row. We call these things raphan. Okay, raphan molecules. They are probably what are called mucopolysaccharides. Okay.
Okay.
Now each one is represented. I should show you first why we have this. Let's see. Okay, I want you to take a pencil or a pen and stand it on its tip and watch what happens. Let's see, where'd it go? Somewhere in here. Come on. No. Let's see. Oh, here it is. Here's the figure. Okay. If you take a pencil and you balance it on its tip, it'll fall over, right?
Right.
Okay. Now, if it's confined to a one-dimensional space, it will fall left or right. Okay. So we are assuming that these molecules, while they come straight down from, let's call this the membrane, if they come straight down, we assume that they will flop one way or the other. Okay, so that's what our assumptions, that they fall over. Okay, so now going back to my, okay, back to the table here. So these are, the flopping is indicated by +1 or -1. Okay, or in this case by a sloped line, sloping to the left or sloping to the right. Okay?
Okay.
Okay, so we assume that each site has one of these states, or it could be empty. That's what the 0 is, an empty site. Okay? Then we calculate what's called the run length. Okay, so the run length, there are 7 of these in a row. There are 2 in a row. There are 2 zeros in a row, 3 -1s in a row. And we number these runs. Okay, so this is our basic data on the what we call the configuration of the molecules. Okay, of the ray-fan molecules. And then we have the rest of the table is filled with numbers, which are the rates of possible events. Now, if the site is empty, then one of these ray-fan molecules can come in from the cell membrane. And so these will be positive numbers for numbers 7, 8, and 9. Okay? If the cell is, if it's already full, we assume that nothing can come in, but that 3 other kinds of events can happen. That either the whole group can move, let's take this one, number 10. The whole group can move left, or it can move right, or it can hydrate and leave, and leaves, its site will become empty. Okay, so there are three rules here. And we assume that these events are what are called concerted. In other words, that a group can move just as fast as a single particle. Okay? And there are a number of examples in the real world of concerted, what's called concerted diffusion. Okay, so that's the basic part, that they move, groups of them will move together. Now, let's see. So this indicates a full site. The ray fan can hydrate to the left, and if it goes to the left, it pushes the diatom an increment to the right, and it becomes empty. If it's already empty, then it can be filled. If it's full and it's tilted this way, then it can hydrate to the right, and it pushes the diatom in the opposite direction to the left, and it also becomes empty. Okay? Okay. So this is sort of our symbolic arrangement. Now we have one other problem, that sometimes you can get a mixed group like this. We assume that the mixed groups can rapidly all become the same. And we assume that there's a bias involved, which is possibly introduced by an electric field inside the cell that would cause them to flip. See, being molecules, they can just do it by what's called rotational Brownian motion. Okay? And for now, we're not simulating that. We're just assuming it happens very rapidly. Okay? Cooperative rotational Brownian motion. Okay? Okay, now, so what we do is we have a step called concatenation, where these groups become consolidated one way or the other with a bias assumed that if there's an electric field. Okay, now let's see. Okay, this is just a curious thing which is not explained yet. This is the speed of diatoms versus temperature, and you can see it falls off. And I make a reference to a very funny paper on similar curves for the flight of houseflies. If you put a bunch of flies in a box and you heat it up, at some point they stop flying. You can imagine why. Okay, and it's a paper by Fitz-Looney.
Yeah.
It's a great paper. Okay, so let me go back here. Okay. Oops. Put it up here. Don't need that. Save it. Let's see. I don't need that. Oops, what's going on here? Why am I not getting my paper back? Window. No window. I must have closed it somehow. Okay, let me reopen it. This one.
Yeah.
No, that's not it. File, open, recent. Oh, come on. Oh, there it is. No, that's not it. Yeah, I lost track of it. Oh well. Where lost track of it? Let me go back to the original and reopen it. Okay, here we are. Okay, so this is yet unexplained, but you can imagine that you'd have to include the mechanisms of cell death to figure it out. Okay. At any rate, so it's a funny observation. Yeah. Okay, let's see. Did the balance here? Okay, now there's a peculiar thing about diatoms, and that is when they move, they seem to move at a constant speed. Okay, so in order to explain that, we use dominoes. If you set up a bunch of dominoes and knock them over, it's been shown that except for a brief transient, the speed at which the diatoms move is pretty constant, the speed at which they fall over. Okay. So we're assuming, see, the energy that for this falling over is, of course, gravitational. But suppose this represents a molecule that hydrates. You can imagine that a molecule hydrates, and that triggers the hydration of the next molecule, which triggers the next one, etc. Okay, so we assume this in the model, and that it's a domino. It's basically a domino theory of the hydration of these molecules coming out of the cell, which then come out of the RAFE. Okay, and that way we can explain, or at least simulate, motion at a nearly constant speed. Now here, let's see. This is okay. If you plot the known data on speeds of diatoms, average or maximum versus the size of the diatom, or what's basically the length of the RAFE, you get these lines here, and both of them are below what's regarded as significant for being different from no correlation with length. Okay. So, because you have a mystery. The diatoms, as they reproduce, they get smaller and smaller, and yet for a given species, it will go at the same speed no matter how long the cell is. Okay, and that seems to be a general, but perhaps not universal, but a pretty general observation. Okay, so we use the domino theory then for the propulsion. So here's a configuration of RAFE and molecules in the RAFE. If they're tilted this way, you get forward propulsion of the diatom, and if they're tilted this way, you get backwards propulsion. Okay? See, nobody's come up with an explanation for backwards propulsion, but when a diatom is moving, as the movie said, it moves in a jerky fashion. It's not simply jerky; it actually goes backwards. Okay? I can show you that. Let's see. This paper just came out. Kinematics. This was done with mostly with John Sabunco. And let's see, way down here. No, that's not it. That's angular. Let's see. This might be. Oh, here. Okay. So if you look at the distribution of the displacement of a diatom from one frame to another in the movies, and we did movies at about 890 frames per second. Okay. you can see that you get negative displacements, not just positive ones. Okay, so the diatom is moving in a jerky fashion, but it keeps going backwards a little bit.
Right.
Okay.
Is that inertia?
It goes forward, backwards. Sort of like the old saying, 3 steps forward, 2 back.
Yeah.
Yeah. Okay. Okay, so let's go back here. Okay, now what does the swelling do? Well, if you start here with, this is representing the RAFE, and this is the diatom trail. If we start in this configuration, it looks like each RAFE, when at least if you have a high-speed diatom, high speed means about 30 microns per second. Okay, if you have a high-speed diatom, then for each one that comes out, the diatom moves 3 units in the opposite direction. We don't have that, which is indicated by this change in configuration. So I've got three units here. If another one comes out, there's 6 units, etc. Okay? So you'd end up here with the RAFE empty and a long trail.
Okay.
Okay? However, while this is coming out, we had to simulate that it's also filling from the membrane into the RAFE. So it's like having a set of dominoes fall over where you can set up as fast as they fall.
Okay.
Okay, so that's a bit different from the human situation. Okay. Now, one of the things that's kind of neat here is if you look at, these are Wolfram patterns developed by an assortment of his rules, and some of them are what are called chaotic patterns. Okay, like
yes.
Rule 150 maybe, or or Rule 30. Okay, so what we have here, you see, the way these things are done is these are each row of these matrices are consecutive states based on Wolfram rules. What we've done is, oops, gotta go way down here for a second. If we look at these things the same way, if these are consecutive states, dark representing the RAFE and in the RAFE, then you get something a little bit similar to a Wolfram pattern, chaotic type pattern. And here you can see, here it is with the tilt included. Now the bias allows the tilt to keep changing, but in this case I think it's, yeah, bias is 0.8. So most of the time it's tilted right, so the diatoms moves left, but occasionally it's tilted the other way, so the diatom will move in the opposite direction. Okay, so this shows, here's one flipping and then finally hydrates. Let's see, over here is one that diffused 1 unit over here. We only have 50 events simulated here, so there's 1 change between each line. Okay.
Yeah.
Okay, so this program took us a couple of months to debug, by the way, and Shruti and Krishna did a hell of a job.
Oh, yeah, I imagine.
I gave her a middle name after the new Martian rover, Persistence.
Oh, yeah.
Okay, because we had a hell of a time thinking like a molecule and making sure the code matched the thinking. Okay.
Yeah.
Okay, now let me show you some of the results.
Okay.
Let's see. Oh shoot! Didn't go back to it again. What on earth is going on here? File, open, recent. There it is. No. Nope. It disappeared on me. Okay, I gotta reopen it. I don't know what's going on with Word here. Okay, here we are. We're back here now. So let me show you some results.
Okay.
Let's see where they go. Okay. Yeah, here. Okay, now here's an example where there's no bias. So the diatom keeps moving forward and backward and forward and backward, backward, forward, 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 backward, back. Okay? Now one thing we're doing is we include a gradient, sort of like this, that falls off exponentially, so that most of the RAFN comes on the left side. So despite that, this is pretty erratic. But if you make the bias increase, you start getting motion mostly in one direction. Here's 0.6. And this is only 50 events. So here's 0.7, goes forward, and then the whole thing reverses. Okay. Here's the bias of 0.8, and now you can see it's going forward most of the time, but occasionally it goes backwards. Okay, this is probably a more realistic one. And then if you make the bias completely, let's see, this is 0.9. Okay, 0.9 does pretty much the same going backwards, forwards. So there's a general average velocity of the diatom, but occasional motion backwards. And if you make the bias 1, then it's all forward. Okay. So we think that qualitatively we have simulated the jerky motion based on molecules.
All right. Yeah.
Okay. But we're not sure. But there's so many free parameters that one can make an enormous number of different models from them.
Right.
Okay.
Yeah.
Okay. So that's where we're at.
That's wonderful.
Okay. I will.
Yeah.
Thanks for presenting that. That's,
sure.
Very interesting.
Okay.
Yeah. Do you have questions, Susan?
I'm just going to say I have to leave a bit early, but
Okay.
that's okay.
Okay. At any rate, it was a real slog, but we finally did it.
That's great. Yeah. So I have some questions.
And,
go ahead.
Okay, sure.
Oh yeah. So
Go ahead, ask your question.
Yeah. So I'm interested in this information processing of diatoms, and so the slide or the graphs you showed at the end there, I think, and then also the symbolic model where you've worked out the different states of forward, backward, and empty. That kind of fits into that because it looks like it has a flavor of information theory in it, and also
maybe.
Right. Yeah.
Well, it certainly, if you ignore the orientation of the molecules, it's a 2-bit system. That's what I did for the Wolfram pattern.
Yeah.
Okay. And then it's just a bit pattern that changes with time. Now, one of the things which Sabunko found when we did the other paper, is that you can fit the curve of a moving diatom very accurately with what's called diffusion with drift. Okay, but the drift is substantial, which gives the diatom generally moving in one direction. All diatoms move one direction, then they eventually reverse. We don't know what makes it reverse. We're speculating there's an electric field that inside the diatom that can reverse, but we don't know. Nobody's looked for it. Okay? So our attitude about the simulation is that it approximately produces the jerky behavior of the diatom. It suggests that there might be a bias of some sort, and we're suggesting the bias might be electrical. That the molecules are charged and that there's electrical bias. Okay. I think one of the novel things here is the introduction of the domino theory, which leads to constant motion, constant speed, except for the jerkiness. Okay?
Yeah.
And then the concerted diffusion allows whole groups to move at once. We're not sure how important that is. That would require further playing with the program or enhancing it.
Yeah.
Okay, we're going to publish the program, so anybody who wants to modify it can do it.
yeah, that'd be great.
Okay.
What is known about the electrophysiology of diatoms? Is there a lot known, or
no? That's a little bit of evidence is known that it exists, but it's basically not explored.
Okay. Wow. Yeah.
Okay. And nobody has attempted to correlate with motion. Nobody's attempted to put diatoms in an electric field, an external electric field, to see if that biases their motion. I mean, these are obvious things to do, but they haven't been done.
Yeah, I kind of noticed that. I was working on that paper on the psychophysics of diatoms, and I was looking for some stuff on that, and there really wasn't anything. But yeah, that's interesting work.
Well, one of the mysteries of Basilaria is that all reports of Basilaria movement is that it is smooth. Right. It's not jerky. And I don't have an explanation for that. Okay. However, it means that it's also worth looking at Basilaria at very high speed to see if there is any jerkiness at any temporal resolution. Okay, nobody's done that experiment.
Diatoms respond to light and will move toward.
Oh yes, yeah, and presumably yes. There's
That's an argument for an electrical connection.
Yes, yes, that's people discuss that, but the mechanism is totally unknown.
Oh.
Okay. There's a lot of discussion of that in our new book, which just came out. Okay, let's see. Let's see. Yeah. Okay. Okay, here's the book. Let me share the screen again. Share the screen. Let's see. Can you read it or larger?
I can't see the screen share yet.
Oh, okay. Oh, it would help if I turned on sharing. Okay.
Yeah.
Ah!
There we go.
I got an infinite. Okay. Can you see it now?
Yeah, the code. Yeah.
Yeah, 24. Yeah. Yeah, this book was just published.
Right.
Okay. Okay, and Stan Cohen's got a lot of information that he's put in it on the response of diatoms to light and whatnot, and a little bit of discussion of the it might imply an electric field, but no measurements yet. Either external or trying to stick an electrode in them, I suppose.
Right.
Okay, nothing.
Yeah.
Okay, so at any rate, if you know anybody with a high-speed camera who will play with Basilaria, it would be well worth taking pictures at high speed of Basilaria. You don't have to get the whole colony. You do it close up for a short time at high speed to see if the speed is indeed smooth or jerky.
Right.
Okay, I think that's a critical thing about Basilaria. I'm not sure if any of the models and the parameters of the model would allow smooth motion or not. I mean, we went for jerky motion and we got it. Okay.
Yeah.
But we had to we introduced these novelties of concerted motion, concerted diffusion, concerted hydration, and the domino effect. Okay.
Right. So, I wonder if the reason Basilaria is smooth is because it's embedded in the colonies that you have. I mean, is your model of jerkiness for single cells or?
Oh, of course. Yes, ours is for one RAFE on one side of one cell.
Okay, yeah.
Yes. And we assume everything's in a straight line.
Okay.
Some RAFEs are curved, and the diatoms end up going in circles.
Oh, yeah.
Literally.
Because they follow the curve, and it just kind of takes them around.
Yeah, yeah.
Wow.
Okay. So we assumed that the geometry of the RAFE was irrelevant to the jerkiness. And so and in writing the paper, we tried to write down all of our assumptions so people could challenge them, or use different assumptions and modify the computer simulation.
Right. Okay.
And the key to keeping it straight was indeed that table. The table includes all possible events for any given configuration. So once the RAFE is in a given configuration, you calculate all the rates for all the possible events, and then you pick one of those events at random. They're not picked uniformly, they're picked in random proportional to the rate counts that you assign to the different events. Okay?
Yeah.
And then you execute that event, you change the configuration, you do it all over again. So it's a lot of computing, but it didn't slow. It's very fast anyway, at least for only 50. We did 50 so it would fit across the page.
Right.
Okay.
Yeah.
Okay. A real diatom will have around 1,500.
Okay. Yeah. Yeah.
Okay. Okay. So that's why I say it's you know, the simulation seems to work, but it's really just the sampling of the possibilities.
Right.
You know, if somebody wants to explore either what we did or to change it, we'd be glad to give you the program.
Okay. Yeah, that's great. Yeah, we'll follow up on that. And yeah.
Because obviously it needs a lot of work. Like, you know, how does the diatom move for different parameters?
Right.
Okay, we only changed the bias.
Right.
And the bias represents an electric field, the strength of an electric field in a given direction. Okay,
Right.
as I said, it ranges from 0.5 for no bias to 1 for full bias.
All right.
Okay.
Yeah.
Okay. At any rate, we only got it in a couple of months late.
Oh yeah, yeah. But that's good. Yeah, that's.
Hell of a debugging effort because, you know, we'd simulate it, the configuration to say no, molecules don't do that. Back to the code, figure out it was usually an indexing problem somewhere.
Yeah, yeah.
or something out of order.
Yeah.
Like we'd have stupid things like a print statement of the configuration before it changed.
Right, right.
Okay, so it was a hell of a debugging effort.
Yeah,
we kept flipping the program back and forth between India and Canada, puzzling over it.
Yeah.
And then we seem to have finally got it.
Well, that's
Okay?
good. Yeah.
So at least I hope the reviewers don't find any errors.
Yeah. Yeah.
We're tired of debugging this program.
Oh, well, congratulations on the paper. It's really interesting.
Okay.
Yeah.
Yeah, so, you know, we're not saying that this is the mechanism. We're just saying it's a possible mechanism, and it's the first attempt to explain it at a molecular level. Now, by the way, one reason we did this is because in reviewing this stuff, and if you look at that volume on diatom gliding locomotion, motility, there are a couple of papers which state very strongly that there's no evidence that the molecules inside the cell are linked to the diatom trail. The standard model is that there's a chain of molecules going from actin to myosin to the membrane to a membrane protein to the RAFAN to the externals, and these RAFAN molecules are dragged along by the myosin molecules, which are the motor molecules. That's a standard model which most people believe, and there's absolutely no evidence for it. Other than if you destroy actin, it stops. But see, in our model, that's why I showed you the bubble at first. In our model, what we think the actin is doing is cytoplasmic streaming, which causes these narrow channels of flow in the cell membrane, which then directs the RAFAN to the RAFE. Okay? And therefore, the speed of the diatom can be similar to, but it can also be quite different from the speed of cytoplasmic streaming caused by the bundles of actin. You saw the bundles mentioned by Pickett Heaps in the movie. By the way, Pickett Heaps did a lot of beautiful work. That's a segment of a beautiful movie he made, and we're trying to get permission to get it up online now because it was a commercial product. And he did beautiful work. Unfortunately, he died last year.
Oh. Yeah.
Okay. So,
Yeah.
let's see. Have I stopped? Oh, yeah, it did stop. Okay, cancel. Yeah, okay. Okay, turn it back to you.
All right. Well, yeah, thanks a lot for that. And I look forward to the program. And
Sure.
if you're watching here, the program will make it available to people who in the Slack and other places. So, I guess.
Yeah, we can give you a copy.
Okay. Yeah, that'd be good. That'd be good.
Okay.
Yeah, no problem.
Okay, I'll ask Shruti to do that. Send you a copy to post.
Yeah. That'd be good.
Okay.
All right.
And as I said, just runs in Python, which I guess is available on most computers now.
Yeah.
I just ran my first program in Python.
Oh. I haven't run any.
Well, it's hampering me. All these things like machine learning, they want to do program in Python, so I'm deciding to learn it. It's very C-like, so it's not a problem.
You know, this program is very simple in its construct, and that it's really just a whole bunch of nested ifs and thens and whatnot. Okay, and loops. So it's structured. You could write this in Fortran too if you wanted. For some reason, writing it in Mathematica, we found extremely difficult to debug.
Yeah.
And they got so frustrated, they took the Mathematica program and rewrote it in Python. And even then, it was still hard to debug.
Yeah.
Okay.
Okay. Yeah, that's great.
Okay.
Thanks. Okay, I'm gonna move to some more. Well, I'm gonna move to some papers now. I know we have this queue of papers that have been mounting, and I like to go through them because it gives us kind of a taste of different things that are coming out in the literature. And
could you send me the references, because I have a presentation to Dr. Sharif I have to do in 8 minutes.
Okay, yeah. You go ahead. I'll catch up with you in email. So, okay.
Thank you.
Good luck.
Thank you. Nice seeing both of you.
Nice seeing you. Yeah.
More people need to turn up.
Yeah.
Okay. Oh well.
Okay.
Bye.
Bye. Let me share my screen here. And so, yeah, we have a couple of papers here. Let's see. The first one is this paper, The Design Principles of Discrete Turing Pattern Systems. This is from Journal of Theoretical Biology. So we've talked about Turing patterns before. We have the Turing reaction diffusion model, and it generates patterns through the activity of the model. So the idea is you have different gradients of chemicals and their boundaries, and those boundaries are, when those gradients kind of overlap, you get boundaries, and then you get things like striping or other types of patterns in chemical systems. And this is something we see a lot in morphogenesis. It's one possible model of morphogenesis, but this is something that Turing actually developed independently of his computer science work. So, so in this case, they're talking about design.
Yeah, it was his last.
What was that?
It was his last paper before the Brits killed him.
Oh yeah. So the abstract says that the formation of spatial structures lies at the heart of developmental processes. So they view spatial aspects of development as key here. However, many of the underlying gene regulatory and biochemical processes remain poorly understood. And so this is where you have Turing patterns that are a main candidate for this process. But Turing patterns, in the way that are described in the original paper, appear sensitive to fluctuations and variations in kinetic parameters. So if you look at naturally evolved systems, how do you expect a Turing pattern to form there? I mean, they form spontaneously to some extent, but there's also a genetic control aspect of it. So you get, and even if it forms spontaneously, you can't ensure that you're going to get a consistent result. And in a lot of animals where you have striping, or you have something like in the Drosophila embryo where you get stripes, or in other contexts, those stripes are usually pretty consistent in terms of how they come out. They're not wildly different from organism to organism. So this is how, we have to wonder how these things evolve. If there's a striping pattern that evolves, how this remains consistent. So the vast majority of mathematical studies of Turing patterns have used continuous models specified in terms of partial differential equations. So, you have a set of partial differential equations, and you can describe patterns. But those kind of models aren't really amenable to necessarily to the biology. I mean, they're just kind of abstractions of the biology, saying this is what is going on, but it doesn't map to, say, some specific mechanism. So here we complement this work by studying Turing patterns using discrete cellular automata models. So now they're using, and I know we've talked about using cellular automata to look at different patterns on seashells or on other things. So here they're actually replacing the partial differential equation using these discrete cellular automata models. We perform a large-scale study in all possible two-species networks and find the same Turing pattern producing networks as in the continuous framework. In contrast to continuous models, which are the equations, we find that these Turing pattern topologies to be substantially more robust to changes in the parameters. So in other words, when you have changes in parameters of the model, biochemical noise or other fluctuations, those things are going to, this model is going to be robust to those. We also find that diffusion-driven instabilities are substantially weaker predictors for Turing patterns in our discrete modeling framework. So diffusion-driven instabilities are things like noise or things that happen when you have, things like turbulence. There's a lot of these kind of instabilities that occur. And so you want to be robust to those. And so these are actually weaker predictors in their discrete modeling framework. So in other words, these cellular automata are robust to those kinds of things that drive change. And they're more robust in the sense that the presence of an instability does not guarantee a pattern emerging in simulations. So we show that a more refined criterion constitutes a stronger predictor. So this similarity of results for the two modeling frameworks suggests a deeper underlying principle of Turing mechanisms in nature. So in other words, this is they're talking about this equation model, and this is one way that people have been able to approximate these Turing patterns. And there's this other model, which is the cellular automata model, which in a lot of ways is more similar to the biology because you have cells that are interacting. And they're saying that the cellular automata is more robust, but also that these models converge, so that it's really maybe kind of driving or sort of uncovering some sort of underlying set of mechanisms. So we don't know which one is the right answer. We assume that the more robust model is the right answer, but it's definitely
okay, Bradley.
Yeah.
Look at what I put in the chat.
Okay. Oh, your paper on from 2011 on the dilemmas of hierarchical instabilities. This is the ones in.
Yeah, the problem I ran into is that when you have, say, Turing models are usually used for cell differentiation.
Right.
And if you try to apply it twice, it doesn't work.
Okay.
In other words, if a cell differentiates into another cell type, and then that differentiates into more cell types, you get what I called hierarchical instabilities.
Okay.
The Turing model just can't explain the structure of an organism. It's great for a single step of differentiation sometimes, as a possible explanation, but the other thing is, there's one of the big examples people like to bring out is the marine angelfish, which has all sorts of intricate patterns on its surface. And people who studied it have shown that the cells are migrating, producing these patterns. That they're not Turing mechanisms, even though they might match Turing mechanisms.
Yeah.
So the whole idea of Turing morphogenesis may be a red herring.
Yeah.
Okay.
Yeah. Yeah.
Anyway, I know people keep pursuing it because it's a neat mathematical trick, but.
Right. But it's so yeah. And I guess this is it's a nice also visual example where you can see the sort of self. Well, last week we talked about self-organization. So
it's sort of like, it's like the snail stuff, you know. Snail patterns look like Wolfram patterns sometimes.
Right.
Okay, and therefore people assume they are Wolfram type patterns, but there's no proof.
Right.
You know, and I can give you an analogy. If you look at dried mud, it forms beautiful cracking patterns. But would you use that as an explanation for the patterns on the back of a turtle?
No.
Probably not. Okay, so, you know, I'm skeptical about all of these. Oh, we make a pattern, it looks like this pattern, therefore it must be.
Right.
You know, no, there are other possibilities in the universe. Okay.
Yeah.
So.
Yeah, that's good, and that's
At any rate, I don't know. You might want to look into it some more. See if I'm wrong. See if anyone's found anything where they proved that a Turing mechanism is actually operating.
Yeah, I don't think anyone's done that yet. So yeah.
Yeah, well, it's been a long time. It's been 70 years.
Yeah.
No, 69 to be precise. Okay.
Yeah.
Okay.
Here's another paper. This is from the bioRxiv. I don't know if it's been published in a journal yet, but this is the theory of branching morphogenesis by local interactions and global guidance. And so the abstract on this reads, branching morphogenesis governs the formation of many organs such as lung, kidney, and the neurovascular system. So when they say branching morphogenesis, they mean it's a ubiquitous developmental process where a number of morphogenetic events cooperate to give rise to complex tree-like morphologies. So in this case, you're looking at like different types of branching within the in the organ. So you know you have things like the lung, where you have air passageways that are almost fractal that go down to the edges where they're very small, or vascular systems. So this is very broadly applicable to neurons and vascular systems, tracheal systems. So many studies.
I got into that. I got into that early when I refereed a couple of papers on branching patterns of trees.
Oh, okay.
Back from the 1970s. I guess it'll be late 60s or early 70s.
Oh, okay.
Yeah.
Many studies have explored system-specific molecular and cellular regulatory mechanisms, as well as self-organizing rules underlying branching morphogenesis. So again, we're back to the self-organizing rules aspect, and you know we could describe that as a Turing pattern or a Turing process, but in this case, they're looking at branching morphogenesis specifically. So in this case, they're looking at a different kind of model. In addition to local cues, branch tissue growth can also be influenced by global guidance. So now we have these local cues between cells, like you might see in a cellular automata. But you also have this global guidance, which is where this tissue is branching, and there's, you know, like I said, sometimes it's fractal in nature. So you start with large branches, and then they become progressively smaller. And so that requires some global guidance, you know, not something that's like conscious or supervised by, you know, the entire organ, but just something that is beyond like just small groups of cells. So here we develop a theoretical framework for a stochastic self-organized branching process in the presence of external cues. Combining analytical theory with numeric simulations, we predict differential signatures of global versus local regulatory mechanisms on the branching pattern. So these are things such as angle distribution, domain size, and space-filling efficiencies. So this is almost like an energetics type theory. So there, I can't remember the author, but there's a theory of sort of branching in energetics, and I'll have to pull up the reference maybe for next week. But you know, these are like, this is a common thing in physics to look at. But anyways, we find that the branch alignment follows a generic scaling law determined by the strength of global guidance, with local interactions influencing the tissue density, but not its overall territory. So you're looking at like these local interactions determining how dense the tissue is in certain places, but the overall territory or the sort of almost like it's not really positional information per se, but it's like a map of that positional information is the overall territory. So this is, we talked about positional information last week as well, and this is where you have each cell kind of knows where it is in relation to other cells. And so, you know, it's not really clear what that global coordination is, but this is something that needs to happen in terms of a territory. Finally, using zebrafish innervation as a model system, we test these key features of the experiment or model experimentally. Our work thus provides quantitative predictions to disentangle the role of different types of cues in shaping branch structures across scales. So again, you know, some of these global guidance mechanisms, you know, could be things like chemical gradients, or they could be electrical fields. I know, I think it was maybe 2 weeks ago, we talked about the flatworm. And what's interesting about the flatworm is you can take a single cell out of a flatworm and you can put it in a dish, and that single cell will reconstruct the entire worm. So each cell is totipotent in that it generates an entirely new organism. And one of the... what was that?
An egg will do that too.
Okay, yeah. And so one of the things that Michael Levin has proposed is that flatworm, there's an electrical field that's generated by that cell, and it gives global information. So as that cell is sort of dividing and filling up space to make a new worm, it's being guided by these electrochemical cues. So that's one way of sort of coordinating globally. And again, it's defining that territory. What is the worm supposed to look like from a single cell? And we don't know about eggs or single germ cells, but we know that, you know, they can also do this where they're totipotent and they can produce an entire organism from a single cell.
Bradley,
So, yeah.
I put a small book in the chat by Robert Rosen, which includes, I think, a chapter on things like optimal branching angles and stuff like that.
Yeah, optimality principles. I know this book. Yeah, thank you.
Oh, you do? Okay.
Yeah, I've come across
Sure.
that. Yeah. Yeah. So that's, and then there's another author, and I can't remember his name. Oh, Adrian Bahan. He's an engineer, and he's come up with some things in that similar vein where he's looking at a lot of branching systems. Yeah. So, you know, and so this is a somewhat. Oh, go ahead.
Yeah. Okay.
Yeah.
Can you send that one to me? Because I'm looking into this local global question.
Yeah, no problem. Yeah, I'll send it. Yeah, so this is a graph here where so this is about they're talking about like these different fields and how branching proceeds across them. So you start off at a single point and you get branching, and there's increasing self-avoidance. So these branches kind of know that they are supposed to avoid themselves and not get tangled up. The field strength increase. As the field strength increases, you get sort of this more sort of regular branching, kind of like a fan. In this case, it's more randomized, and so you can see that this field strength, whatever this is represented by.
By the way, you can get similar thinking. I don't know about modeling yet of branching structures in centric diatoms.
Okay.
Which are much more regular than these patterns, but they are branching patterns.
All right. Yeah.
Okay, and
that would be great. Yeah.
okay. Yeah, just look up. Just do a picture search on centric diatoms, and you'll see that some of them have these kinds of patterns.
Yeah. I'll look it up. Yeah. Finally, let's see, which paper do I want to do here? How about this one? This is Actually, this is a soft matter paper, so this might be something Susan will be interested in. But I'll do it now, and I'll send her the paper. This is nice. I guess in soft matter they have these nice images here on the front cover.
Yeah.
Fingering instability and spreading epithelial monolayers. So this is going towards the formation of the epithelium, which is part of the embryo. Roles of cell polarization, substrate friction, and contractile stresses. So this is where we're looking at the formation of a sort of a tissue layer, and it's basically talking about the physics of this process. So
I see. They're making it similar to dendritic growth of crystals.
Yeah, I think so. So the abstract reads, collective cell migration plays a crucial role in many developmental processes that underlie morphogenesis. So now they're focusing on collective cell migration, the movement of cells collectively. So if you have like morphogenesis, of course, wound healing is another example where you get cells that move towards the wound and sort of seal up, and they form this fibrous layer that protects you from bleeding out, or your your blood system from getting infected, or cancer progression, which is of course something where cancer cells are invading different parts of the body. In such coordinated behaviors, cells are organized in coherent structures and actively migrate to serve different biological purposes. So they form structures when they migrate, and this is what they're getting at here. In some contexts, namely during epithelial wound healing, so in this case, specific case, it is well known that a migrating free edge monolayer develops finger-like instabilities. So that means that as they move forward, the front edge forms these fingers that sort of project out of the main group of the main front, and they form instabilities because they're not, they don't remain like constant. They're constantly seeking out in different directions. Yet the onset of these is still under debate. Now, here by means of theory and numerical simulations, we shed light on the main mechanisms driving the instability process, analyzing the linear and nonlinear dynamics of a continuum compressible polar fluid. So they're looking at this in the context of a fluid.
Stupid question. Does this paper mention Rayleigh instabilities?
They may, down in the paper.
R-E-Y-L-E-I-G-H, I think.
Yeah. I don't think I can search this particular, in this, when I have it up like this, but I'm not seeing it in here so far. But I can send you the paper.
Because usually when you have periodic instabilities like these, they're attributed to. Do you know what Rayleigh instability is?
Yeah, I'm kind of familiar with it.
Where a cylinder of fluid breaks up into drops.
Right.
You can watch it just by slowly running a kitchen tap.
Right, yeah.
You see, by the time it falls, it breaks into drops.
Yeah.
Okay, and that would be my first assumption in anything like this. That these are Rayleigh instabilities, and they're initiated at a molecular level. Variations at a molecular level is presumably.
Right. So this is a migrating epithelial monolayer. If we have if we look at the cells here, so these are moving in this direction, and you have all these different types of physical parameters in your model. So they have pneumatic length, which you mentioned is this sort of type of organization, and there's a length to it. Shear viscosity, traction forces, cell substrate friction, intercellular contractility, which is how the cells are contracting against one another, and then the surface tension coefficient. So
basically all of these occur in various forms. If you do a Rayleigh instability on a surface, you end up with traction. You end up with traction forces.
Okay, yeah, that's T.
A simple way to observe it is take some honey, liquid honey, and take a tiny bit of it and spread it in a line. You can watch it happen.
Oh, yeah.
Okay.
Yeah. It's
okay. It's very, very easy to see it happen, and it would have most of these characteristics.
Right. Let's see. So they get into the linear theory shows that it is crucial to analyze the perturbation transient dynamics. And so we unravel a plethora of crossovers between differential exponential growth rates during the linear regime. The numerical simulations suggest cell-substrate friction could be the mechanism responsible for the formation of complex finger-like structures at the edge, since it triggers secondary fingering instabilities and tip splitting phenomena. So,
Okay.
it's kind of like this branching where you get things that come up, and then there are other sub-processes. So, so these things are sort of not, I wouldn't want to say fractal, but I guess they are, but they're very itinerant, so they don't.
No, they actually have a characteristic wavelength
Oh,
rather than
okay.
being fractal. If you look at the early literature on the Rayleigh instabilities, they didn't predict the long-range shape, but they predicted that the perturbation grew exponentially.
Okay. Yeah.
Okay, because they couldn't handle the nonlinearity of it actually forming drops.
Right.
But that's probably been overcome in the fluid literature now.
Yeah.
Okay.
Okay. Finally, we obtain a.
So it's kind of
Oh.
yeah. This kind of paper is kind of rediscovering well-known physics.
Yeah. Yeah. And they're applying it to the biological system.
Yeah.
Yeah.
Whoopie-doo.
Yeah. Finally, we can obtain a critical contractile stress that depends on cell substrate friction and the initial to nematic length ratio, characterizing an active wetting to wetting transition. So this is, this is a lot of physics that they're just applying to this model. And we've talked about this with soft materials and how, we can learn from some of the stuff that's going on in soft materials and how they apply to biological systems. And this is an open debate as to whether this is how useful this is, because on one level it's an analogy, and you're just trying to fit things into the model that are biological. So
Yeah, and of course there's active energy generation, which you don't have in a drop and in liquids, or ordinarily.
yeah,
Yeah.
I don't see any mention of a Rayleigh instability in here, but
Yeah, probably not. Probably not. Like I said, it's a rediscovery
yeah,
of stuff that's 150 years old.
yeah,
Okay.
yeah.
But see, this is a general problem in biology. If a reference is older than 3 years, it's irrelevant.
Right. Yeah. No one reads it. Yeah. Yeah.
So, okay, have a good day.
Yeah. Have a good week. Thank you
Take
for
care.
attending.